Good morning, I'm Bruce Campbell. Welcome to the first PNOC informational webinar for 2022. As many of you know, May is National Brain Tumor Awareness Month here in the US. Today, our panelists will talk to you about the latest biological findings and treatment options for DMG DIPG.
Hi, and I'm Alan Campbell, and we co-founded PNOC Foundation in 2013, 3 years after the diagnosis of our son's brain tumor. PNOC Foundation is a nonprofit public foundation really set up to create funding and awareness and support for PNOC's researchers, clinicians, and specialists.
Today, you'll hear from well-renowned doctors specializing in DMG, DIPG, about the latest or the current treatment options, clinical trials, and biological findings specific to this tumor type.
And you'll also meet a lovely patient family dealing with the disease. We really want to hear from all of you today. Today is really about information sharing. So please put your questions in the chat and we'll be fielding those and hopefully answering everybody's questions.
If you're joining us for the first time, the Pacific Pediatric Neuro-Oncology Consortium, or PNOC, is a leading global pediatric brain cancer consortium with over 35 study sites in the US, Europe, Australia, India, and Israel, collaborating to improve treatment outcomes in a unique way and translating the latest biological findings in brain tumors to clinical trials and more effective treatments. A key mechanism in this translational research are the unique PNOC working groups, which match investigators from around the world with specific disease type, expertise, and focus, and through rigorous scientific collaboration and consensus, the most promising ideas are developed into clinical trials. Advocates and philanthropists are also invited to participate and lend their partnership.
Let's introduce some of these panelists, distinguished panelists that we have today. I have the honor of introducing Dr. Sabine Mueller. She's the PNOC co-founder and project leader. She's an assistant professor of clinical neurology at UCSF. We have Dr. Karl Cashman, Joan and David Evans Pediatric Brain Tumor Scholar, assistant professor of pediatric neuro-oncology at Chad Carr Pediatric Brain Tumor Center, Michigan, and Dr. Javad Nazarine, head of DMG Research Center of University Children's Hospital in Zurich, Switzerland, associate professor of pediatric Children's National Hospital in Washington, DC. And last but not least, we also have Rachel Casales, one of our team members from PNOC Foundation. She will be helping with fielding some of the questions from your Q&A.
Great. So let's turn it over to our panelists to give us an overview of the tumor type. And I think what we're going to cover off today for those who joined us last year is what's changed in the last 12 months. So looking forward to a really interesting and updated discussion.
Yeah, thank you very much, Bruce and Alan, for the introduction. And it's really like what Alan outlined. It's really, this is information for you. We are really trying to answer all of your questions. We thought we will start with just walking through what we think is sort of best practices when you are first diagnosed, to give you a little bit of our vision of this, how this should look like. And then really Javad and Karl will also talk a little bit about some of the trials we have happened and what we could expect next. And then we will hopefully open this for discussion. So I think any family faced with this disease, and these are tumors that are located in the pons, which is sort of a part in your brain that is really managing a lot of functions, controlling your how you move your arms, your legs, your breathing, your eyes, your swallowing. This is when we call these tumors DIPG, Diffuse Intrinsic Pontine Gliomas. And over the last years, we learned that tumors also occurring in other midline structures also have shared biology. So now we are often using the term diffuse midline gliomas, and that normally includes DIPGs. Sometimes the terminology can be confusing. But I think my number one recommendation, or our number one recommendations as you start this journey when you are faced with this disease, is really build a team, get support, accept help. Can I advance or you have to? As I'm sure those who have been faced with this disease, you know, the next discussion normally that occurs with your team is the radiation therapy. That remains today our standard of treatment. I think we are learning that there are different subtypes of these diffuse midline glioma, and some actually might not much benefit from radiation therapy, but to date, it still remains our number one treatment recommendation after the initial diagnosis, because most children will benefit. Often their initial symptoms when they are presenting will get better, and they then will undergo sort of this, what we call a honeymoon period, or a period where really the symptoms can almost completely disappear and children's overall feel better. There are now new therapies that are being explored versus a conventional schedule of radiation therapy is about 30 days. So most children get radiation therapy Monday through Friday, and it's about 5-6 weeks. But we are now exploring, could this be shortened? And this is sort of a hypofractionate schedule where it's maybe only 20 times a child receives radiations, or even 15 times. So these are things we are currently exploring in clinical trials. There are certain countries where it's more standardized, that it's already the shorter schedule, but I think there's still a lot we need to learn. And then obviously there's always the discussion protons versus photons. Based on our discussions we had with many groups, I think that there's no strong recommendation either way. So we generally don't recommend to our families, if for whatever reason the site has no protons available, or it would take longer, we would normally not recommend protons for this disease. There are indications, and I think it's always worth a discussion when you discuss it with your radiation oncologist. But in general, conventional photons is considered still the standard for these tumors. I think a big discussion that every family should have is the use of steroids. I think those who have children who have been started on steroids, they know that steroids can have very positive effects in improving the symptoms. They work very quickly, and it's a very safe medication. The problem is, if you use the steroids long-term, children can have significant weight gain. So it's something to be discussed if that's something really you want to continue long-term. And more recently, we learn about the use of bevacizumab, or another term is Avastin, where sometimes when children experience some swelling and inflammation as they are undergoing radiation therapy, maybe there are alternatives now that are available instead of using steroids to find alternatives. Next slide. And then one of the key questions is always, should a biopsy be performed when and where? So I think a biopsy, that is our recommendation, and I think it should be done in centers who have neurosurgeons who are experienced with this procedure. In an experienced neurosurgeon's hands, I think that is a fairly safe surgery. Most of the children tolerate this very well, and there are many now of even published studies showing that. I think the biopsy can be performed before or after radiation therapy. We have shown safety in both settings. I think it would really help with the diagnosis. As I mentioned earlier, there's definitely differences we see in these diffuse midline gliomas, DIPG, and for some clinical trials now, these are targeting very specific alterations or changes we see in the tumors. And unless we know that a child has that specific change, we cannot enroll them into the trial, because the therapy would just not work. So I do think it's important. The next step is often that families and parents are faced with this participation in a clinical trial. There are now many options patients have, especially in the United States. There are many trials when you go on clinicaltrials.gov, which is a great website where you can search by disease term. You see many, many different trials. Sometimes it's really hard to understand if a trial is actually open, if your child is eligible for the trial, can you enroll in the trial? What does it really mean to participate in a trial? And this is where really it comes back. You have to have a team that they can advise you and guide you through this. It's often helping reaching out to the enrolling trial site to get an understanding what this would really mean for your family to participate in the trial. Some trials start with the initial diagnosis, even before the biopsy is taken. Some trials start before radiation therapy. But there are also many trials now that start after radiation therapy, and I will outline some of our trials when they will start. And then I think something we often hear from families, getting a second opinion. Will this upset my team? Is that something I can do? And I would strongly recommend to get a second opinion. If you feel like that's something you like to do, I think everyone has the permission in my mind to go and seek a second opinion. Sometimes what we see that happens is that families reach out to many different institutions, and then you get different opinions, and then sometimes families feel pretty lost with all the different advice they get. So I think that's something to be careful about. And then also there's certain financial burden sometimes where insurance might not cover all the different needs. At this point, my strong recommendation is talk to your team, talk to your social worker. Normally there's always an option. Next slide. Other resources outside sort of, the active treatment, they are wonderful in most hospital and centers. Child life resources you have, palliative care teams, they're often a helpful resource even in the beginning of the disease process to help really a family cope with everything. They provide emotional support. And then really a great ally is your social worker who provides support not only for the patient, for the siblings, but also for the parents. So now changing a little bit from this initial status to, what you should do in terms of clinical care, about some of the trials we developed as part of our Diffuse Midline Glioma adaptive combinatorial trial. This is what DMG Act stands for. And what we decided to do, which is a little bit different compared to other trials, that really as part of this, what we call a platform trial, wanted to offer therapies at different stages, because we heard from families that often they're diagnosed, everything happens very rapidly, and then they are not eligible for a certain trial, or they missed the enrollment phase for that trial. So we really wanted to develop a trial where we can bring in patients into the trial at different disease stages. So cohort one is for newly diagnosed patients with diffuse midline glioma. You can have it or not have a biopsy. Patients after completion of radiation therapy, and then patients with evidence of progressions who are eligible for a second round of radiation therapy. And down here we're listed all my great collaborators and supporters for this trial. But next slide. And so based on some wonderful preclinical data that really Javad and Karl has been leading, we designed this trial. So it's based on using ONC201, which is a medicine that is taken orally and has shown in definitely a subset of these diffuse midline gliomas to have success, and then combine it with different agents. And so the two combinations we put forward is basically panobinostat and paxilosib. As part of the trial, we wanted to also understand how much of the drug is getting into the brain tumor tissue. And this is what we call the target validation study. So patients are scheduled for their standard of care biopsy. Prior to the collection, they receive one of these agents, and then they move on to radiation therapy. They get one of these agents with radiation therapy, and then will be randomized to the combination. And so we have started this trial end of last year, and it was really a great accomplishment having that platform trial, which allows us also to open and close arm rapidly based on preclinical data and clinical outcome data. And as soon as we opened the trial, unfortunately, the company who supported, the arm with panobinostat, unfortunately, had to close their support. So right now, this is the arms we have in the DMG Act trial, PNOC 22. So, and then as mentioned earlier, you can basically enter the trial at different disease stages. We can collect biopsy at different disease stages. And we are collecting really a lot of correlative studies that really hopefully will help us to identify markers that can help us to predict who are the children who will respond to this therapy, who are the children who will not benefit from this therapy, so that we can use something different. And that includes blood collection, the tumor tissue, cerebral spinal fluid, and most recently through the support from really the Children's Hospital of Philadelphia, but also our colleagues at the Princess Maxima Center in the Netherlands, the microbiome. So even stool collection, where we have seen in other studies that changes in stool can really help us to identify potential biomarkers. And this is another trial that originated out of this really combined working group that Bruce and Alan introduced about diffuse midline gliomas, is ONC206. So ONC206 is a sister molecule of ONC201, but for that, we don't know the dose yet. So we are starting basically identifying the safe dose that can be given to children. Once we have that, then it's a very similar to design than what I just mentioned, where children can enter when they're newly diagnosed, post-radiation, and at time of progression. Upcoming concept within PNOC, one of the hurdles, and we can discuss this later, is this intact blood-brain barrier to really get in sufficient concentrations to the brain tumor. And so there are several methods that are being used to overcome this. And we have used convection enhanced delivery in the past, where catheters are put directly into the brainstem and the therapy is being delivered. But now we are also exploring the use of focused ultrasound to disrupt the blood-brain barrier, but also for photodynamic therapy. And then obviously immunotherapy, great trials ongoing at multiple centers in multiple different consortia, including Stanford, Seattle Children's Hospital, Texas Children's Hospital, amongst others. The one we will move forward is a very specific directed against the H3.3 mutation that is present in the majority of these tumors. And we hope to launch this trial this summer. And with that, I will hand over to Javad.
Great. So I thought to give some overview of some of the work that we do on the preclinical space and how the drug, what processes they go through in order to make it into the clinic. Two examples of what Sabina mentioned were ONC201 and ONC206. As most of you know, we typically start with thousands of drugs that may have a good indication for other type of cancers. We then test these drugs in vitro. This means the cell lines. Sabina mentioned some of the specimens that we collect during the trial. They may include cell lines that are collected from a biopsy, and that gives us an opportunity to develop these cell lines. And these are model in vitro, in petri dish models of the tumor that we study. We have now built a zebrafish facility where we can test the toxicity of such drugs with the idea of looking at the neurotoxicity or the toxicities that the drug may have for the development of an organ. Of course, a drug may be really good in killing tumor cells. At the same time, it could be toxic to the healthy organs where they're developing. And if the drug passes all these three steps that we have, then we take them to in vivo testing, where we test the drug in tumor-bearing mice. A lot of this work is being done in the laboratories of Karl Cashman, our lab in DC, UCSF, and with our other colleagues that I will mention. But essentially, this is a very long process. It's a very hefty process, and it takes really the collaboration of multiple laboratories to be able to streamline. The platform that we have built with the PNOC has enabled us to do this. These are some of the collaborators that I wanted to just give the credit to highlight the fact that the two trials that Sabina mentioned, PNOC 22 and 23, with ONC201 and 206, are really the result of some of the colleagues that you see here from Australia to Europe to US that share data, specimen, and resources readily without publication with the sole focus of developing trials that work. We have now developed a platform where we have the largest number of DIPG, DMG cell lines that are being characterized. What you are seeing on the left is the microscopic images of the cell lines and what are the proteins, different proteins that are expressing. In red, for example, is nestin, in green is GFAP. telling you that these are glial cells. We are characterizing these cells, sharing them among different laboratories, and again, developing a platform where data and resources flow rapidly in order to develop clinical trials. We have developed specialized centers that you see here, that again, Australia, Europe, and US. These are specialized core that have specific interest and entities and expertise. Pathology, for example, in vivo that I mentioned to you, drug screen and PD markers that are being done in Karl's lab, UCSF in Sabina's lab, and our lab, and others. Data management at CBTN, computational biology that is being done at Oslo, proteomics, etc. There are multiple needs that we have in order to take the best drugs to the clinic, but also when Sabina mentions the correlative studies to look at the drug response to see if the drug is actually interacting with the targets, and or if there's a resistance that develops after drug is administered. All of those are being studied in the preclinical studies or studies that we do in the context of correlative studies. The cores that we have highlighted here are some of the few cores that are highly engaged in such studies. With that, I will hand this to Karl Cashman, who will present some of the data. Karl?
Hi, thanks. Happy to be here, and thanks to so many of you for joining. So this is something that we didn't cover last year, but is an exciting update. We looked at the results from the phase 1, where for many of the patients who got treated with ONC201, they got baseline sequencing of the tumor. And so what I mean is patients who enrolled on ONC201 phase 1, at the time they were diagnosed, their tumor was usually biopsied. And we took some of those samples and enrolled patients on a program at U of M called MyOncoSeq. This, to this date, is a research study. So it's CLIA certified, so it can be used in the clinic, and it can be used to confirm mutations as well as to act on those mutations. But it's not something that's billed to insurance. It's done with a consent form upfront, and the results are returned to families. So when we opened this trial, we knew we wanted to have a really progressive approach to having molecular data paired with response to these various arms. When we went through the various options, MyOncoSeq seemed like a good fit because we had done a fair amount of this paired sequencing with how did patients respond to this drug in particular at U of M. And so this was chosen as the sequencing tool we would use. And fortunately, the leads for this sequencing at U of M were really enthusiastic to participate in this. And what we now have as part of when patients are enrolled on either PNOC 22 or 23, they're consented to the study, but they are also asked, would you like to have your tumor sent for sequencing? And that can be performed through the same pipeline with no additional cost to family. The main thing that we have to think about is the amount and availability of tissue. Go ahead and advance, Javon. So this was as of maybe a month ago that there were about 16 enrollments on PNOC 23, which is the phase 1 of ONC206, and 17 enrollments on PNOC 22, which is the DMG Act trial. And we were excited to see that a majority of these cases did sequencing as part of their enrollment. And what became very apparent, and as many families are aware, with this tumor, when you biopsy it, there's not an infinite supply of tissue. We estimate that for most biopsies, there's somewhere between 3 and 10 cores taken, and those cores are a little smaller than a grain of rice. And often by the time patients may enroll in these trials, there's little to no tissue left. So we're very aware of that. And actually, one of the things we wanted to do for these patients is, in addition to the panel sequencing, which just means searching for the key genes that we know are clinically relevant and may push families or clinicians to one treatment or another, that panel sequencing is done through MyOncoSeq, but we also wanted to do whole genome sequencing, which is really looking at the entire DNA portfolio for a tumor and for the patient's healthy cells. And we want to have all this information for each of the patients treated with these drugs so we can know if they're responding. Is there something in the DNA or RNA that might help give us an understanding of why patients are responding or not responding, and what might be a good future combination. And so one of the things that we worked with our group was to say, when you're sequencing for the panel, can you save the same sample that you're using for the panel and feasibly use it for whole genome and not use any additional tissue? And our lab has been able to do that, and the first waves of whole genome sequencing data is being provided. It doesn't provide what we call really deep sequencing, where you can do hundreds of copies of each part of the genome, but it probably will give us enough data to ask and answer the questions we want without using any additional tissue. So we're really excited about that. The funding for this has been coordinated for the first few years through Chad Tuff Defeat DIPG and the Musella Foundation, and a recent addition from the Head for the Cure Foundation. And we are, as part of this, it was very important to us that the data became available to researchers and collaborators immediately. And so we're in the process of transferring over the data to Children's Brain Tumor Network, which is the largest brain tumor consortium, largest brain tumor data repository. And they are going to be able to make that data very available, and also in a sort of searchable, usable way that clinicians can easily look in real time. So that depositing is ongoing. Go ahead, Javad. And then this is just sort of the leading edge of the correlate research. We've talked in the past perhaps about some of the things we can find in liquid samples for these patients. And so my lab and other labs, including Javad's and other labs around the world that participate in the DMG Act working group, are developing sort of the next generation of correlate tests. And we have a really progressive design in this trial about collecting spinal fluid and plasma in a safe but frequent way. And we're just in the process of vetting some of these tests that you see here, and we don't have time or really I don't know if this is the right group to look through all these, but just so you know, we do think that we're going to be able to learn more and more information from these liquid samples from patients, and again, learn how patients are responding or not responding to the drugs in real time. For the most part, this will be done as a group and batched so that we can learn to help inform the future trials. But if we have data, obviously, that's really impactful and potentially clinically relevant, that may be something that we can circle back to the treating clinicians. Go ahead. So this is a list of all the funders, and probably actually we're missing some. So we apologize if you recently supported this effort and we haven't gotten your group on here. Please let us know. But it's a huge community, and we certainly wouldn't be making the progress we are without the support from this group. This is really a crowdsourced project from the families and foundations that were most affected by this tumor, and we're excited to get after this tumor with the support of this community, many of whom are on this call today. And so we'll stop there, and we've left hopefully a really healthy chunk of time for discussion. We knew from the last call that was actually some of the best part of this webinar at the past time.
Rachel or Sabine, do you want to pull up a few questions, and then we could determine which, or the panelists could determine which of the ones best to answer it? There's some that were in there.
Yeah, so there were a few questions about, do international patients also have access to our trials? We are trying to open these internationally. As I mentioned, we are about to open more sites in the United States. We are hopefully opening up these studies in the Netherlands, as well as Israel and Switzerland, our sites in Europe. Right now, for a patient to participate in the PNOC 22 trial, you have to come, unfortunately, to the US. Not every center in the US accepts international patients, but we have successfully enrolled international patients in the trial. but that's a big commitment and a big discussion. So, but it is feasible, but that would we have to discuss the details further. I think there was an overall question. I think, Karl, maybe I will let you answer this, about what we currently know about the effectiveness, efficacy of ONC201 as a single agent in these tumors.
Yeah, so just to clarify, the ONC201 in particular finished, actually it still has a large and many-armed phase 1. And typically we wouldn't use phase 1 data to help answer the question that's being asked. And so the phase 2 studies that we're doing, at least the DMG Act, also add other drugs. But the benefit of that phase 1 was that pretty early on, those of us involved with that trial learned that we could move up to the weight-adjusted dose that we would move forward as the phase 2 dose moving forward. And so a majority of patients on that phase 1 actually got the phase 2 dose, but the trial wasn't designed to fully answer the question about efficacy. That being said, we did see a strong signal. The data we have, and we've shared some of this in abstract form at a number of conferences, and we're hoping to publish it this year, shows that in K27M, if you look at patients who got ONC201 either post-radiation, before recurrence, or at recurrence, in general, it was very statistically significant, a benefit compared to historical controls. And this is really exciting because I think for us, it's the drug that has given the most consistent signal. We do know, and we've discussed in this meeting and in the presentations we've given, that it appears that patients who are treated post-radiation before recurrence seem to have the best effect in that phase 1. And as well, there's a small benefit for having a K27M tumor in the thalamus compared to the brainstem. But we also know there's still a significant number of patients who don't seem to have benefit from the drug. And we still don't know why that is. And I think we're hoping to ask and answer that question in these follow-up studies, and also hopefully to gain further ground with the combination, or hopefully with ONC206 as well.
Great, thank you. There was one question from the audience asking the difference between the design of PNOC 22 and how that compares to GBM Agile. So GBM Agile is a truly also sort of based on bringing multiple arms in for adult patients with glioblastoma, which is also very challenging to treat brain cancer. But statistical design is different. So what the methods our trial is using is sort of a Bayesian design that will allow us to bore information from the different arms to then at a certain threshold either drop an arm or basically add in arms based on the data that the preclinical DMG Act team is really generating. So there are differences. It goes into a lot of details about the detailed changes of the statistical designs, but same concept where you basically evaluate different arms and then bring them in and out. Any other questions?
Yeah, there was a question about the disruption of panobinostat.
So why that? Yeah, so I think that is hard for me as an investigator to fully answer. I think that was a company decisions. I mean, the company was it wasn't based on that there was additional toxicity data or that they were not excited about a trial. It was just a company decision that they cannot support this any further. So that's all I know about this. But it had nothing to do that there was more data was available that would be concerning of toxicity. That was just unfortunately stopped.
Mm.
One question that I think is a question for Dr. Nazarian. The question is, when you evaluate the toxicity of a specific drug in your preclinical models, what is really the timeline associated, and is toxicity evaluated over the life of a patient or the development of a particular drug? So maybe we can answer it in two ways. We can start.
Sure. So at the preclinical level, we usually, when we test a drug, we are looking at the developing embryo. This is usually zebrafish, because all of the organs are developing and the development is very similar to that of human. The idea is that if there is a toxicity associated with a specific organ, we can detect that in a developing embryo. There are other different modalities also of looking at toxicity. One could be behavior. We can also look at the changes of behavior of a developing embryo, zebrafish, that may be affected by the toxicity of the drug. The second aspect of it is the neurotoxicity. We can dissect the embryo and look at the potential neurotoxicity of a drug. And if a particular drug that is good in killing tumor cells, DMG cells, passes all these different steps, meaning that it has low toxicity by affecting the organs in less degree at the dose that is effective in killing the DIPG tumor cells, we then pass this drug to the clinic. And then the clinical toxicity of that is something, a different aspect that Sabina will explain.
Yeah, from a clinical side, how we assess toxicity, especially if we start like ONC206, where we really don't know anything about the dose. We started as a dose that we know was basically safe in our animals. And then there are certain formulations, what is then a good starting dose for that in patients. And then we normally, what we call, have sort of this 4-week period, what we call the dose limiting toxicity period. So we assess patients very frequently, every week in clinic, we check their blood, and depending on other toxicity profiles, we do additional safety studies. And then that assessment, if for the first 4 weeks a patient is doing quite well, then we basically go to monthly assessment. But it doesn't mean, and then as part of a trial, the study team reviews every week all the adverse events that are being reported from patients, because for some studies we learned, or for some drugs we learned that actually initially they were well tolerated, but then as patients were taking these drugs longer, there was more toxicity. So it's a continuous evaluation, but the first 4 weeks being sort of our key determined if you can also increase a dose level or not. But that is basically how we do it. But it's really throughout the course of being on a clinical trial, the toxicity is being monitored, and the study team is monitoring every patient through every week. I don't know, Karl, if you have anything to add to that toxicity.
No, you guys nailed it.
We have another question here about any indication of efficacy of ONC201 against H3 wild type tumor overexpressing EZHIP.
That's a really good question. And I think the short answer, unfortunately, is we don't know. There were very few to no patients that have been treated in a way that we could assess for that. But we purposefully designed DMG Act and actually the PNOC 23 with ONC206 to include these patients, to not exclude the H3 wild type DMGs. And that statistically, we would actually have to separate out these groups because they have different survival outcomes, because it was such a big unanswered questions, and we didn't want to leave this relatively rare sub-patient population aside, not knowing whether or not there was a signal there as well. So I think for right now, the answer is we don't know, and that we think there's enough of a possibility that there will be response in this group that they're included in this trial.
We just have to add, it's the same at the
Javad, I think your volume cut out midway.
Sorry, can you hear me now? Good. I was going to say that the preclinical level is the same, because we don't, the number of cell lines that we have that are wild type and overexpressing EZHIP are small. They're responding the same, but the number is not large enough to make that statistical decision.
Okay.
So yeah, then there were 2 questions. So one is, again, maybe for Karl and Javad, how often are we successful in establishing cell lines from biopsy? Maybe we can start with that question.
Yeah, I think in Zurich, we've been able to develop cell lines 6 out of 10 times, 60% of the time we've been able to develop cell lines. It really depends on the proximity of the surgical units to the laboratory that is capable of doing this rapidly.
Yeah, and I think, you know.
Oh, go ahead, Sabina.
No, you go.
I was just going to say, it's variable between sites. So first of all, almost any site, even if they're doing biopsy, they most likely are not generating a cell line. There really needs to be a lab there with a PI with the experience. I mean, it's Javad's lab is able to do it, but most sites are not generating cell lines. One of the issues we have at our own institution here is that the policy for biopsies for brain surgeries is to not use any for research, that it has to be done through a consented clinical trial and clear clinical benefit for the extra tissue. One of the ways that we're getting around that, and it's an idea we actually have learned from this DMG working group, is a possibility of using what's called a needle wash, where this is generally discarded debris. When a surgeon goes in, collects tissue, and then washes the needle before going in again, that debris is thrown away, and we've convinced our own institutional research board that that's safe for us to use, and we've had some success starting cell lines from that. But it's not something that is in widespread use unless there's a really active investigator at the site.
And I would add to this that I think, I mean, we are often successful in maybe with the initial cell line development, but then often after you try to really develop them, then they often don't continue to grow. So I think depending what really the cell line definition is, I mean, and the numbers you see published in terms of success and what you hear is really ranges between 30 to 50 to 60%, and obviously it depends also a little bit on the subtype. There are other questions about, and we get this question quite often, you know, if you cannot participate in the trial, can you get, for example, paxilosib outside a clinical trial? And so the access to investigational agents outside a clinical trial is through what we call compassionate care request, which a lot of companies support. However, if there's a clinical trial open, then often there is no support for this anymore. So it really depends on the agent and where the company stands to that. And then for the clinical team also, not every institution, I would say, can accommodate those, because then basically what these compassionate care requests often are, you have to write a protocol for that specific patient, you have to get a contract in place. So it's a lot of regulatory work, even if I would say the review can happen quite quickly, but it is a pretty big lift to get this in place. And it often sort of depends if a team has the capability of doing that, but also if a company is willing to provide that. Other questions is about side effects for paxilosib. There's a question if hypoglycemia has been seen. I would actually say it is a known side effect of paxilosib. We have had few patients so far, but it has not been a major problem for us. And then we would use agents like metformin for the treatment. If metformin independently obviously is beneficial, that is still under investigation. Karl, there was a question, maybe you can answer, if you can speak to the advancement in treating DIPG, which loss of trimethylation, but EGFR alterations.
Yeah, this is sort of similar to the previous question. We now know that a small but steady number of patients with midline glioma, specifically in the thalamus, either with or without H3K27M. We've seen either with the EZIP overexpression or H3K27M or H3 wild type and no EZIP. Any one of those three, we've seen EGFR alterations, or we've seen just very high EGFR expression in some of those patients. It's a minority of thalamic tumors. And I think this is a group that our early signal from the phase 1 showed the strongest resistance to ONC201. And again, the phase 1 was not designed to fully ask and answer questions like that. So it's just an early concept. But it is one that we will hope to learn more about in the current studies. Personally, when we see patients with this, I do talk to them about that and how this may not be the most optimal phase 2 trial to join. We know that there's not great options for EGFR inhibitors that get into the brain and have pediatric data. And so this is a project that my lab is working a lot on. And the Yuval Tuwari Foundation, which has been supportive of a number of labs in this working group, has supported us to work on working with drug companies to find an EGFR inhibitor and seeing how it works for this small but real patient population where ONC201 by itself may not work well in the cases we saw so far.
Okay, at this point, I think I love hearing about all of this and how it relates to real patients. And I just wanted to take an opportunity to introduce Mateo Lambasa. I had the sincere pleasure of meeting her virtually. She's got a very amazing story about her family's long journey from Croatia to here to California. And Mateo, I was wondering if you could share with our group, just so that other patient families could really, hear some of about what you're going through with Chiara and the trial that you're on. I think that would be really helpful to
Yes,
them.
of course. I want to say hello to everyone and thank you again, Alan, for calling me to assist the panel today. Our journey was truly long. We came here all the way from Croatia, so we traveled halfway through the world to get some treatment options for our Chiara, because me and my husband, and probably as any family who's going through this horrible disease, giving up wasn't an option for us. It's truly hard for people from Europe, especially from people from Croatia, to get good cooperation with your local doctors when you get a disease like DIPG or DMG. We had truly the most amazing local oncologist back home in Croatia who told us, please do everything what is in your possibility to get Chiara some way of a treatment and to get her hope. Because when you hear a diagnosis like this in Europe, especially in Croatia, the local doctors, most of them tell you, enjoy the 6 of 9 months you have with your child. It's only palliative care. And I think it's a sentence that none of the parents who are going through this want to ever hear in their life. That's when me and my husband decided to search for clinical trials through clinicaltrials.gov, the website. It was truly a hard way. It was a long way, a hard way. It was night and nights on our laptops, sending emails all over the world. We were lost. We didn't knew anything about this disease. But the only thing we knew is that we wanted to get her some kind of treatment options and hope. I know that the current trial we are on isn't a cure. I'm aware of that. But I know it's hope to prolong her life and to get at one point at the future to a cure and to let our Chiara live and grow with us, with her family.
Thank you so much for sharing that. And we're thinking of you, and it's hard not to be touched by that.
It's truly hard for every family who is going through this, but in my opinion, please never give up on your child. Do everything what is in your power. If you hear is impossible, there is no cure, there is no way, there is always something. Listen and look at your own child. It's a hard way. We have been through a hard way as well. We had a little girl who was diagnosed almost a year ago in July last year. A little girl who has gone through everything without any pain, without any problems. A little girl who could walk on her own 2 months ago. Now we have a little girl who is in a wheelchair, who isn't able to walk on her own anymore. But we always have faith. We always have faith, and we are blessed when we see her and when we see other children in worse situations who are going through this same disease. So please, for everyone, always keep hope, always keep faith. There is always something that is going to help you. Maybe not the same cure or treatment for everyone, but I truly, truly hope that somewhere in the future, at one point, with these great doctors who are working on this every day, every single day of their life, who are devoted to this disease, that they are going to find something that is going to be a cure for everyone in the future. I know it's harder for European families to come to the United States, but you can always do locally fundraising as we did. You can go through local media, you can go to GoFundMe platforms to raise the money which is going to get you to the United States, because local European insurance give their hands up and they say it's all to you. We cannot afford to pay you anything. Never give up. Never, never, please. To everyone, keep faith, and something I believe is going to happen. I hope the sooner the better for everyone. It's a hard way. It's a way for the whole family, not only for the child. It's hard for the parents, for the siblings, for everyone to see this. It's hard, especially when you are at the other side of the world, apart from your family, apart from your friends. But have joy, have faith in every day, and believe your doctors. Together, you truly can do everything. Be open-minded to them. Speak to them as you speak to anyone else. And together you can do a lot. Thank you, everyone, for listening.
Mateo, thank you so much for your strength, your courage, your optimism. That's really, really inspiring. Thank you for joining us today.
You're welcome. Thank you.
Do we want to revert to a couple more questions that I don't think we covered in the chat? I think we covered this one off, but maybe not specifically to this question. Thanks, Karl. Are you investigating ultrasound to disrupt blood-brain barrier with EGFR inhibitors?
That's a great idea. We're not that far ahead, although as you may have picked up on, the various groups in the working group may combine those concepts together down the road. We have two really good leads on drugs that target EGFR and get into the brain well. So we're hopeful that they will prove to work well with ONC201 and well in some of these midline gliomas and not need further alteration to get across the blood-brain barrier. But it's a nice tool to have in the future in our panel to think about ultrasound with any of these drugs. So it's not an unreasonable idea.
Look, and I think that's one of the things that's really unique about this group is just, how collaborative it is. And that includes, as I said in the opening, with patient families, philanthropists and patient advocates. It's this sharing of ideas in events like this, which are truly unique. There's another one here. Has there been any new studies with AMXT1501 in combination with DFMO? Is that combination available in a clinical trial?
Yeah, so as I understand it, there is a trial, and I believe it's mostly at sites in Texas. It's on clinicaltrials.gov. It is, I think, actively recruiting patients now. We don't have any, I don't know of any results from that trial yet.
Okay. Another one, do you think the intratumor heterogeneity of DMG will drive how you match ONC201 with other drugs?
I think this is a very complicated question, but a very, very good one, because I think that's one of the things we are struggling with, to really understand the heterogeneity and how to best tackle this, because right now we are doing biopsies, from one area, and then it is hard to assess really the heterogeneity in that setting, and especially since we are not taking tissue when children are taking medicine sort of on treatment biopsy, we are a little bit limited in our assessments, but that's why we are included so many other correlative studies, including CSF, to get at this. But I let it also Javad comment on this, and Karl, their thoughts on how we can address that heterogeneity in these tumors.
You may be on mute, Javad.
Sorry, we see this often when we're trying to develop a cell line from a biopsy. When we look at the cell lines that are developed, they're a heterogeneous group of different cells. And one of the promising results that we have shown is that at least ONC201 in the preclinical setting tries to reset the majority of the cells to become more committed from being less committed, from being more stem-like cells. We're hoping that we would be able to discover more drugs that can push the majority of the cells that are uncommitted, heterogeneous, to more a committed group. But as Sabina said, we don't really understand the complexity of heterogeneity of these cells. One area of research that Karl's lab has been developing and our lab has been developing is to look at the CSF and to look at the plasma and to be able to decipher the genetic mutations that we can obtain from the tumor that is releasing into the microfluid, in order to understand the genetic changes that the tumor cells are adapting, either as a group that are becoming one population that is resistant, or as a heterogeneous group of cells that are changing as a result of the treatment. We're hoping that the liquid biopsy, along with better work with the actual biopsy, will give us a better window of looking into understanding this heterogeneity of the cells.
Here's another one. My son, 19-year-old, was diagnosed with DMG in July 2020. He is on ONC201 since December 2020. There's a tumor regression, but there is a cyst now at the tumor site. How does ONC201 work in this situation? And a follow-up question, any info on the link between ONC201 and cyst formation?
I was hoping Dr. Mueller would have a really good answer for this. These are really hard cases, and I don't have a quick solution or a generalization from ONC201, other that this is a problem, I think, with any trial, that oftentimes we see the cyst growing and the tumor regressing, and it's complicated. Each patient's probably pretty unique and requires discussion with neurosurgery. And my own sense is it's not ONC201 driven, but I guess I can see what Sabine thinks.
Yeah, no, I would agree with you, Karl. I mean, we see cyst formation, we see it just after radiation therapy. We have seen it on many patients prior to ONC201. So I don't get the sense that children on ONC201 are getting more cysts now than we have seen sort of in average. So I really don't think there is a true relationship. And it's a tough problem. So sometimes, we ask our neurosurgeons to drain the cyst, and that can really help with symptoms. But unfortunately, the cyst often refills. Sometimes the surgeon can create a pathway where the cyst is then draining, but it remains a challenging problem. But I personally, although we haven't really studied this in a way, but I don't see any signal that ONC201 is triggering cyst formation. And the question, does ONC201 work against the cyst? I think that is another one that we don't understand. So unclear right now.
What's the success rate of creating a PG cell line from a biopsy tissue?
So we answered, Bruce, we had
Okay.
this discussion before, and I think what we basically said depends on the specimens, how much tumor content is there, and then depending on the expertise and the different laboratories. So it can range between 30, 40 to 60%, but it also depends at times on the different subtype of DIPG.
Got it. Sorry if I repeated that one. A European ONC201, ONC206 similar to United States ONC201, ONC206?
Yeah, so this is a very good question, and I think we cannot really answer it because the ONC201 that is given out in Germany is not regulated in the same way. It's being given out on sort of what we call in Germany individueller Heilversuch, sort of a compassionate care, but it's not undergoing the same regulatory process. At least we don't have knowledge of that. So I think that's a question I can't fully answer. And ONC206, I think that is extremely challenging to give out on compassionate care because we don't really understand the dose that can be safely given, because we know for these medicines, we need to find the right dose. We need to basically monitor these children. So these are questions, unfortunately, we cannot answer at this point.
And a somewhat related question that we might have covered before, but I'll ask it anyway. What's the difference between ONC201 and ONC206?
Yes, so these are sister homolog molecules. They're very similar. I think they're just the original drug was ONC201. ONC206 is a derivative of 201. They both target mitochondria within the living cell. They shut down the mitochondria, which is the center of energy production. Some of our studies have shown that ONC206 maybe binds more effectively to the targets than ONC201 does. But I'm sure there's more about the two drugs that we don't understand at the cellular level. We don't know why one drug works better than the other in a different setting. But again, essentially they're homolog sister molecules. They have a very, very similar structure, except a few carbon chains that are different.
And then a question from the audience was, it was a more general questions about the DMG working group, what the focus will be going forward and what are the barriers for progress. So right now, really, and we just actually prior to this webinar had a meeting, we are really focusing on developing the next combination arms for these trials, and we are investigating lots of different molecules. And I think the process that Dr. Nazarian outlined, you could see how much work goes into this. We are testing multiple cell lines in different laboratories. We are testing toxicity, but then the really tedious work starts when we test our different combination studies in mice. And that requires many people in the lab working almost around the clock to get any data, but that is really data we need to understand how we can move this best forward into the clinic. And so when it comes to barriers, I think the barriers are, as always, unfortunately, I think, the support, the financial support to hire enough people to do this quicker and faster. The barriers is access to these new medicines. Some agents look very promising for us, but the industry partner might not be really interested. So I would say these are the two key barriers, because we have a highly motivated group, I think, of investigators around. I think we built a great infrastructure, how we could move something quickly into the clinic. And I think if you have support and if you have a motivated industry partner, I think we can translate our findings really rapidly now into the clinic. But I think we remain limited by basically sort of core funding for these more boring but very needed sort of in vivo studies. But I let Karl and Javad also chime in where they see the biggest barriers.
I was just gonna comment, not necessarily on the barriers, but the future steps. Our working group has really changed the way we think about research. Someone commented about the collaboration and the different skill sets And I think we're all learning sort of a newer model of how science can be approached and in a very translational and collaborative way. And some of the things that have come out of this, which we're really excited about, is how various groups have come up with techniques that other groups had never heard of and otherwise left to their devices may never have heard of. And so to speed that crosstalk, we've started a program of transferring trainees across labs, across international boundaries. And I have, for example, a graduate student from a lab in Spain who's teaching us a mouse model. And my understanding is he's next going to be in Javad's lab. And we'll be sending researchers between the sites to try to speed up that and accelerate that process. And Javad's also working on a sort of a mentorship for junior faculty. So we're really able to move the discovery and the pace of research just through the communication between the sites, which we've never really done before.
I can add one more item is that I think in the past 2.5 years, our group has come a long way, as Karl mentioned and Sabina mentioned. We do have some really good clues of what may be a good combination with UNC. Karl has been doing a fantastic job with some of the GFR inhibitors. We have a lot of work to do on the correlative studies because we always get the pushback from clinic as far as as soon as we have a good drug, or what are some of the correlatives of these signatures of the response or resistance. I think we are at a very good point to start to answer these questions, but having dedicated teams that could develop the cores to do such correlative studies, in vivo studies, etc., has still stayed at the paucity, and many people in my lab and Karl's lab and others are doing it as a part of the collaborative team that we have. So that remains a major point.
Great. There was one other question from the audience asking, PNOC has multiple trials and multiple sites. Do all sites offering all trials? How does it work? So that's a great question. So how we structured PNOC is sites can open all the trials if that's what they want to do and what they think they can enroll to, but they don't have to. So some of the trials also are limited, and that sometimes depend on certain expertise. For example, our convection enhanced delivery trials, they're open at limited sites. Most likely the focused ultrasound sites trials will be open at limited sites who has the expertise and equipment. But then in general, we try to really give access to all the sites. It's a different barrier moving these trials, and as we heard so eloquently from our parent today, also bringing trials to Europe is a significant barrier, because often drug companies are supporting maybe drug development in our trials in the US, but then limited to the US sites and don't allow us to expand to our European sites. But we have a very strong focus on this, and this we have successfully opened trials in Zurich, enrolled trials. We just onboarded the Netherlands. We have trials open in Israel. So it is doable, but it is definitely a significant higher bar to open trials internationally. But then each site in general decides if a trial fits their portfolio, if they have a competing trial, and that's normally how it works.
We've got time for a couple more questions. Okay.
So there was a question about ketogenic diet, and that's a really great question, because literally, like I said, just before this call, we discussed that this will be an exciting avenue for us to explore. I think we still don't understand quite the impact ketogenic diet could have. And we are currently trying to model this in our model system, but that is something we are interested in, especially when we are thinking of combining about it with some of our metabolic targeting agents that Javad mentioned.
And then there's a question about the DIPG research and cancer moonshots. Whether or not there's connection. There is a connection. There are fantastic families and foundations in Washington, D.C. and across US that are highly involved in engaging the Congress and to make sure that the funding from the moonshot will actually be dedicated partially at least to pediatric rare cancers, including DIPGs and DMGs.
Okay, so I just want to take the opportunity to thank our panelists. You know, not only are you taking time away from, you know, your research and in many cases seeing patients to participate in something like this, but your willingness to actually answer all the questions, you know, including the difficult ones that might not have answers yet, I think just reflect your passion and care, you know, for what you do. And I'm sure the audience is very grateful. If for some reason we missed answering a question, you can follow up with us post-webinar. I think we were pretty thorough, actually. We got the overwhelming majority of them. I also want to thank the collaborating foundations for supporting the work of PNOC and getting the message out to their networks to, you know, join us today. And it's only through coming together as doctors, as research sites, as scientists, as foundations, as philanthropists, that we're really going to move the needle and make this change.
Yeah, and I'll just add one other thing too, that to your point too, that this is being recorded. So you'll be getting an actual link with how to review this again, as well as a couple other links that Rachel will make sure that gets sent out. So I really want to offer a very special thanks to Mateo Lambas and family for joining us and being part of this particular webinar today.
Thank you again, Ellen. And thanks everyone to let us tell our story to everyone. And another message to all of the families, as I said when I was speaking, please always have faith and hope and never, never give up on your child.
Thanks, Mateo. I think that's a great way to end.
I do too.
Thanks, everyone, for joining us.
Thank you. Bye.
Bye.
Bye, everyone.